real thrill to be here today to talk to you about how things are evolving in understanding human disease using genetic approaches and tools. So what I want to talk about first is how I got interested in science. This is the family I was born into, my mom, dad, and older brother. That's me in the upper right. My dad was, as you can see, was in the Air Force, and I moved around along with the family. We lived all over the United States and abroad. We lived in Ankara, Turkey for a couple of years when I was growing up. And my exposure to science was just through interest in what was going on in school, and I became really interested in the ideas of, I was inspired by President Kennedy in the 1960s to want to do something beyond myself. Medicine seemed like a great outlet, seemed like a very good intersection between science and helping people. And as I became further involved in medicine, it became clear to me that a lot of the limitations in medicine were that we really didn't understand the basic biology of how humans normally work and how disease processes changed the normal biology of humans to cause disease. And so I developed this career of pursuing the interface between basic science and clinical medicine, with the notion that the better we understand the fundamental causes of disease, the better we'll be able to ultimately develop new approaches to the diagnosis, prevention, and treatment of disease. And being a scientist at a great place like Yale is one of the joys that one could possibly have. You get to work with incredibly smart people. These are people who have trained in my laboratory, who have now gone out and run their own laboratory all over the United States and the world. And that's a great thrill and opportunity. And you also get to work with incredibly smart people as a scientist. And the number of people just here at Yale who I've collaborated with over the years is just extraordinary, and involve people in virtually every basic science and clinical department here at the university. And so being a scientist and being a physician is something that just one of the great opportunities to discover something new about how humans work, and that is a profoundly satisfying thing for me. Also get to have a family, and this is my family who have been born and raised here in New Haven, my wife and my 3 kids. And so I want to just underscore the point that there is life in science and outside science, and both are compatible. What I want to start with is how we have come to understand that genetics is important in human biology. And I want to start with the point that as long as humans have been on the planet, we have recognized the fundamental principle that like begets like. And so if you want to have giraffes, you need a male and a female giraffe in order to get new giraffes. If you want to have ostriches, you need to have a male and a female ostrich to produce new ostriches. We have recognized this principle as long as humans have been on the planet. And so we have known something about genetics, that this principle of life begets like is really what underscores the fundamentals of genetics. And we also recognize that this applies not just to animals, but to humans as well. So here is an apocryphal family of 4 pairs of identical twins. And in the absence of genetic variation, identical twins are identical because they share a complete set of the identical chromosomes. They come from the same fertilized egg. And as a consequence, if you look at these 4 pairs of individuals, you will recognize that they don't sort of look like one another. They look indistinguishable from one another. Their overall shape, height, weight, where the weight is distributed is identical, but those are just gross features. They're really fine features of their hair color, their smile. It's virtually identical, and I don't think any of us, if we saw these pairs, would have any difficulty recognizing that these are genetic clones of one another and are virtually identical in their physical appearance. Now, of course, we don't all look like one another. There's tremendous variation in human traits. And this is a beautiful photograph by a famous photographer named Annie Leibovitz that demonstrates this point. This is a jockey, Willie Shoemaker, who rode many winners in the Kentucky Derby, and the basketball player, Wilt Chamberlain, who played center for the Los Angeles Lakers. Now, I don't think any of us in this room would likely believe that even if we are the most ardent proponents of the idea that it's nurture rather than nature that determines the way we end up, I think even in the extreme case, we would have a hard time believing that if Wilt and Willie's parents had exchanged their children at birth, that Willie would have been the one to grow up to play center for the Lakers, and Wilt would have been the one riding horses in the Kentucky Derby. And of course, when we look around this room, or we walk around New Haven on this beautiful fall afternoon, we see that there's tremendous diversity in human appearances. But we also recognize that it's not likely just human appearances that are determined by genes. That genes are also influencing our susceptibility to virtually every disease. Otherwise, how could we explain the paradox of Jim Fixx and Winston Churchill? So Jim Fixx was 5'10" and 150 pounds, Winston Churchill 5'8" and perhaps on a good day 270 pounds. Fixx was a marathon runner. Churchill was slothful by his own description and legendary for his gluttony at the dinner table. So Fixx was also a healthy lifestyle promoter. Churchill was a smoker of fine cigars. And from everything we know about lifestyle, we would have said Fixx is leading the healthy lifestyle, Churchill is leading a lifestyle that should lead to early demise from a variety of ailments. So despite stacking the deck in this fashion, how can we explain that Fixx, in fact, is the one who died at age 52 while running, a cautionary note to those of us in this age group? But Churchill, despite all of these adverse risk factors, lived to the ripe old age of 90. Well, of course, Fixx had something going against him from the outset. And this is something that we all recognize, that diseases frequently, we say, run in families. And what we mean by saying a disease runs in families is that if somebody in your family has a particular disease, you are somewhat more likely than anyone in the general population to also have that disease. And in Fixx's case, he had this genetic predisposition that his father died of a heart attack at a very young age of 43. And so we have suspected from families like this that there are likely to be genetic factors that are contributing to the risk of disease. But until the last 20 years or so, we've had almost no ability to use that information to understand what those underlying causes might be. So how have we gotten to a state where today we can actually begin to understand these problems in human populations? Well, this all dates back to about 150 years ago when Gregor Mendel, a monk in Austria, began an interesting series of experiments of breeding different varieties of peas together. And what he recognized, that there were some very unexpected outcomes of the kinds of breeding that he did. And he found, for example, that if he bred pea plants that only produced round peas and those that only produced wrinkled peas, all of the offspring only produced round peas. But if he then made further breeding between those offspring, he found that reproducibly, 75% of the offspring produced only round peas, but 25% produced wrinkled peas. And he found the same kind of pattern occurring with different traits in his crosses. And this led him to recognize that there were factors, he had no idea what those factors were, that were being transmitted from the parental pea plants to their offspring, that were then segregating as discrete factors, so that, and he deduced that in this case, in this cross, there were two different versions of this factor. We'll call them plus and minus. And if you inherited one or two versions of the plus factor, you always produced round peas. And but only if you had two of the minus versions did you get wrinkled peas. He had no idea what these factors were, but he could deduce that they were being transmitted in an unmodified form from generation after generation. But statistically, 25% of the offspring of a cross where you had one copy of the plus, one copy of the minus allele, you would get offspring that were only wrinkled. So from this, we developed an idea that there were these factors that were being transmitted in an unmutated form from generation to generation, and these were determining the phenotypes, in this case, of these pea plants. The next big step in the evolution of genetics came from the recognition of where these factors resided in the cell. So the cells were known at that time, and it was known that inside the nucleus of cells, there were these things called chromosomes. And it was recognized that the number of chromosomes was different from species to species, and in some animals, the number was different in male and female animals. And this ultimately led Thomas Morgan, a geneticist who worked on fruit flies, to prove that these factors that Mendel had identified resided on chromosomes inside the nuclei of cells. And his pivotal experiment was to cross fruit flies, one that had red eyes, one that had white eyes. And what he found was that in this cross, the white eyes always traveled with the chromosome that was determining male sex. And so as a consequence in this cross, like Mendel's P cross, the first set of offspring all had red eyes, but it was only when you mated these to one another that you produced offspring where there were flies, 75% had red eyes, 25% had white eyes, and the white-eyed flies were always male. And this provided the formal proof that the factor for white eyes was traveling on the chromosomes inside the nuclei of cells. And that was an enormous advance in our understanding, but at that time we still did not know what is it in the chromosomes. We knew that the chromosomes contained DNA and protein. It was commonly thought at that time that the DNA couldn't possibly be the genetic material because there are only 4 different fundamental molecule types in DNA, and there are different bases, and we'll call them A, G, C, and T. And it was thought to be such a simple chemical polymer that it couldn't possibly account for all of the factors that we believed existed in individual species. And the pivotal experiment that proved that DNA, in fact, was the genetic material came from an experiment that Griffith did in the 1920s, where there were 2 strains of bacteria, one of which was virulent and would kill mice when injected into mice, one of which was not. And what Griffith first showed was that you could heat treat and thereby kill the virulent bacteria, but mix them with the non-virulent bacteria and end up killing the mice nonetheless, which that shouldn't have done if it was just infected with the non-virulent bacteria. And if you took the bacteria that came out of that, they were now virulent. So something had been transferred from the virulent bacteria, despite the fact that they were dead, so it had to be some chemical, that got into the non-virulent bacteria and made them virulent. And Avery and others in the 1940s proved that that stuff was DNA. And that proved that DNA, linking all the way back to Mendel, was accounting for the factors that are accounting for transmission of variation in traits from generation to generation. And that then left the mystery of how is it that DNA could possibly provide the code. And Watson and Crick demonstrated that DNA is a double helix shown here that is composed of these 4 versions, A, G, C, and T. And as you unwind this helix, you can see that they're always paired in a way that A is paired with T, G is paired with T in the 2 strands of DNA. And we have learned that this, in fact, is what accounts for how all proteins are made inside the cell. And proteins are the business end of the genome and are what are responsible for all of the day-to-day chemistry and metabolism that occurs inside the cell. So that if this is our double-stranded DNA, A paired with T, G paired with C, this is the library that sits inside the nucleus, and it's copied in various bits by a process called transcription into RNA copies, and the RNA copies provide the code by which all proteins are made inside the cell. And there are ribosomes that are inside the cell that do this translation process. And Tom Stites here at Yale won the Nobel Prize for determining the structure of these ribosomes, that are these proteins that are responsible for making all of the proteins in the cell. So I just want to stop for a moment and make the point of how amazing this entire process is. That these chromosomes sit inside the nucleus, they're replicated generation after generation, and this is the, Watson and Crick recognized that this explained the fundamental mechanism of how is it that we go from generation to generation with our traits being passed in a faithful form to subsequent generations. Because when these two strands are replicated, you separate the two strands, and the sequence of each strand provides the complement of the sequence of the other, and so you can come up with the identical sequence of the two strands simply by separating them and replicating them. And this accounts for how we're capable of passing the same sequence from generation to generation. And this is really a profound insight that has become a fundamental basis for how we think about the consistency of life across generations. You can imagine, however, that occasionally mistakes might be made, so that rather than faithfully replicating this sequence, you might make a mistake. And the replication process is extremely faithful, but on average, there's about one mistake in every 500 million bases. And as a consequence, this is an incredibly faithful process, but sometimes makes errors. And these errors are how variation is introduced into the population and accounts for why we don't all look like one another, but also accounts for our susceptibility to a variety of diseases. I also just want to take a moment to make the point of how amazing it is how chromosomes are wrapped up inside the nucleus of a cell. So in mitosis, when cells are dividing, we have to make precise copies of the chromosomes and then pass one copy of each of the offspring chromosomes onto each daughter cell as cells divide and replicate. And to demonstrate how amazing this folding process is, I'll need some volunteers. So I will think I'll need 9 volunteers from the audience. So who wants to come up and help me? Come on up. Who else? I need some more. Come on, come on, come on. I need a few more. Yeah, please, come on up. Yeah, please come up. Okay, so come up right here. Okay, so let me first, so I'm going to need some more, so come on up. Okay, so this looks like a good group. 1, 2, 3, 4. I need 2 more. Come on up. 1 more now. Just 1. Okay, so I want to make the point that if I picked my finger with a pin and took a drop of blood, I would have 5,000 in a drop of blood, and then that makes about 100 microliters of blood. And in that 100 microliters of blood, there would be about 500,000 white blood cells. And if I took it down to 1 microliter of blood, there would be about 5,000 white blood cells. So this is what you would see in a microscope. And these are the red blood cells, which have no DNA in them, no nuclei. And these are the white blood cells inside a drop of blood. And if I just took 100 cells and unraveled all of the human DNA in those 100 cells, what I have here is the length of what those 100 cells would look. Okay, so I want you to come over here and stand here. And I want you to stand over here. And I want you to do the unspooling. So I want you to take this. I want you to hold this and stand right there. And now I need to do this 2 more times. And so I want somebody to stand here. Why don't you stand here? Okay, and you grab this in, and you the stairs about halfway up. And we'll do the same thing over here. Come on over. Okay, so you hold this, you take the spool, and you run up the stairs halfway. Okay, and now I want to, third person, you come over here. And come further down. Okay, so I want you to take the spool and bring it to her. Come on down, unravel it. And you're going to have to go fast. Okay, and give it to her, and you grab the string, now grab the string, and you go back. I want you to go back and forth until the entire spool is spooled out. I want you to do the same thing.
Oh, jeez. Okay, that's a lot of walking.
So this is how long, and go ahead, and this is the DNA. They'll go back and forth, just back and forth.
Oh, no.
And you're going to have to go fast, because there's a lot of DNA. And so there are 3 billion base pairs in the human genome. And the 3 billion base pairs, when unspooled in 100 cells, comes out to over 600 feet. And it's all wound up inside a single cell that is only 10 microns in length, in diameter. And it's astounding how well that's packed. And in fact, if we took all of the cells in the human genome, in the human body, of which there are perhaps 30 trillion, we would find that the length of DNA laid end to end would extend to the sun and back 300 times. So just think about how amazing the compaction of DNA inside the cell is. How's everybody doing? We have a lot of strands going back and forth, and I think we're going to have to go faster. Everybody's doing an amazing job, and I think we're figuring out just how long this is. And keep in mind, this is just the DNA from 100 cells inside the body. And I think...
Oh, wait, you dropped. Wait, Talia, you dropped one.
Yeah, I dropped one.
And this is where we get to the point where I think we can declare victory and say we have proved our point that the DNA inside our cells is incredibly long. In each cell, in just one cell, it's about 6 feet in length for all of the chromosomes. And when we put it all together for 100 cells, it would be about 600 feet in length, which is the length of these spools. So I think we should say, yeah, it's very long and that you've done an awesome job. Let's give a round of applause. And put it on up here. So just come on down and put it up here. It's great. You guys did great. Thank you very much for your help. So, starting in 1985, there was, oh, you're going to have to unwrap yourself. Thank you. There was an audacious idea that was proposed as a science experiment. And the basic idea was that we would make tremendous progress in understanding biology if we were able to sequence the genome of humans and determine the DNA sequence of all 3 billion bases of the human genome. And that was an audacious idea, in particular because we had no idea how we could actually achieve that goal at the time. But remarkably, this was pulled off in about 15 years. In 2001 and then 2003, a draft of the complete sequence was published. And that gave us the blueprint of what it takes to make a human being. And surprisingly, people had guessed before the 3 billion bases were sequenced, how many protein coding genes would there be in the human genome? And the guesses ranged from a low of about 15,000 to a high of about 120,000, and most people guessed around 50,000 genes. And it turned out that the answer is about 20,000 protein coding genes. But the second part that was really surprising is that as other genomes were sequenced, it turns out that the number of genes and the identity of genes in all vertebrate species, from fish to humans, is pretty much the same number, and they're pretty much the same genes. And as a consequence, this tells us that virtually all genes are conserved. They're present over 400 million years of evolution, and they're they're generally identical in their overall function from species to species, with a few exceptions like gene families that are evolving rapidly, such as the immune system. Well, this led to the question of, if that's true, could we understand how mutations are contributing to disease? And this, of course, assumes that we can find genes, traits that are caused by genetic factors. And the evidence that this is true is that there are rare diseases that recur in families much more often than expected by chance. Sometimes we can find diseases that are prevalent in particular parts of the world where people are all evolved from a small set of common ancestors, so-called genetic isolates. And then sometimes we find traits that are so unusual that they strongly suggest a genetic causation. And then lastly, we can find frequently that diseases are more common in twins who share 100% of their genes than twins who only share, dizygotic twins who only share 50% of their genes. And from this, we can deduce that virtually every human disease has a genetic contribution, sometimes from a single gene whose mutation causes the trait, but more commonly from multiple genetic variants that are likely together causing variation in the trait. And so we see families like this, which would be caused by a mutation in a single copy in a single gene, in which affected individuals transmit the gene to half of their offspring who get the trait, and the individuals who do not get the gene end up remaining free of the trait, and this would be a so-called dominant disease, or recessive diseases where each of the parents carries one copy of the disease-causing mutation, and only if you get 2 mutant copies of the gene do you get the disease, and this would be called a recessive trait. So we have recognized over the years that we can identify these underlying genes, and these can help us understand the biology, not just in the individuals who have the disease, but in some cases can identify mutations and pathways that can be exploited in the general population. And I will give you the example of a disease called Familial Hypercholesterolemia. So this is a disease that is very rare. It affects 1 in a million people, but it causes the affected individuals to have very high cholesterol levels that are exceedingly high compared to the general population. And these individuals are recognized to get heart attacks before the age of 30, something that is very rare in the general population before age 30. And this therefore established a causal relationship between high cholesterol and heart attacks, and led to the recognition that if we could lower cholesterol, we might be able to prevent heart attacks, not just in these patients with this disease, but in everybody. And that has proved to be the case. And we now are using medications commonly called the statins to inhibit the production of cholesterol by the body, with the recognition that this has profound effects to lower cholesterol. This has been particularly impactful for people who are at high risk of heart attacks in the general population. It is a nice example of how we can use genetics to develop new therapies that have impact in the general population. The mutations that cause this disease prevent the normal clearance of cholesterol from the blood through receptors in the liver that normally are involved in clearing cholesterol and sweeping it up from the bloodstream. A second example of how this understanding genetics is leading to new therapies is the first gene that was identified in the contemporary era of genetics. And this is a gene for cystic fibrosis. So cystic fibrosis is a pulmonary disease that causes children who are affected with cystic fibrosis to get chronic infections in their lungs that frequently leads to complications with pneumonia and often leads to death at early ages. The gene for cystic fibrosis was identified in 1989 and was identified to be a chloride channel that is expressed in the cells that line the lung, the epithelium of the lung. And the mutations cause this channel to no longer work properly. And this was an example where the gene was identified, but at the time it was identified, we had absolutely no idea how to mitigate the effects of that mutation. But people continued working on it, and just in the last several years, a company has developed a medicine that can allow patients who have cystic fibrosis where the mutant channel gets to the cell surface but no longer is capable of opening normally. Well, they've identified a drug that is capable of opening these channels, and this has been shown to improve lung function in patients with cystic fibrosis. It is a beautiful example where we had no idea what the underlying cause was. The specific cause was identified, and a medication has been identified that can mitigate the effects of the disease-causing mutation. I'll give another example where the underlying biology has revealed completely new elements that have changed the way we think about a common trait in the population. So we have recognized that obesity is a gene-environment interaction. And we have suspected that there are genes that contribute to obesity. We also know that the availability of calories in the environment is important to the development of obesity. And here are two strains of mice. These are, in fact, two offspring of the same litter of mice. And you can see one normal mouse and one mouse that is a little bit different. It's a little bit different in that it is massive, has massive obesity. And this is all due to accumulation of body fat. And Jeffrey Friedman at Rockefeller University demonstrated that this is caused by a single mutation in a protein called leptin. And leptin is a protein that is involved in, is made in fat cells. And what Jeff showed is that it is made in fat cells and is a hormone that is exported from fat cells to tell the brain how much energy there is, how much fat there is in the body as a source of stored energy. And so the normal function of this pathway is to tell the brain when the fat stores are full, and to tell the brain as a consequence to stop taking in more calories because we've got plenty of stored energy around. And so these mice develop this massive obesity because they are no longer capable of making that protein and sending that signal to the brain to say, stop eating. Well, that applies not just to mice, but also to humans who are missing leptin. So here is a photograph of a 3-year-old and a 7-year-old. This 3-year-old is about 160 pounds at age 3. And if you ask his parents about his dietary habits, they will tell you that they have tried locking the refrigerator, clearing all of the cabinets of food, and the child is just relentless in his drive to eat. And he has achieved this impressive obesity at age 3. He turns out to be genetically deficient for being able to make this hormone leptin. And as a consequence, this profoundly, I think, changes the way we think about obesity, because this is not a matter of will or how willing somebody is to do something. The absence of this hormone is driving his drive to eat, and it is not a matter of his personal responsibility in that regard. Remarkably, this photograph of this 7-year-old is the same individual after he has received leptin for several years. So this one hormone has transformed this child from massive obesity to now having a perfectly normal body habitus. Now, this hormone does not have this effect in the general population, but for these individuals, this is a remarkable finding, and I think has profoundly changed the way we think about obesity in the population. I'll give one other example of, in the case of cancer. So there's a disease called chronic myelogenous leukemia, or CML, which is a cancer that has been uniformly fatal in human populations for as long as we have been on the planet. The course of the disease has been that you're diagnosed with chronic myelogenous leukemia, you'll smolder along for a number of years, and after 6 or 8 years, it kicks off into a much more malignant phase that leads to death. And this was a uniformly fatal disease. It was recognized that there was a chromosome rearrangement that a number of years ago was demonstrated to cause increased activity of a particular protein kinase called ABL. And the ABL kinase is kinases are proteins that transfer phosphate groups onto other molecules. And the activity of this pathway in the white blood cells that cause this leukemia, this leads to increased proliferation of the cells that are involved in this leukemia. And what was done by investigators working with a pharmaceutical company was to develop and show that a small molecule called Gleevec could inhibit this kinase activity and prevent the cell proliferation that results from the activity of this kinase. And when given to patients, it's had the remarkable effect of arresting the development of this leukemia, and patients commonly go into long-term remission, so that while there were only about 6,000 individuals with this disease in the population, there are now tens of thousands of individuals who are surviving chronically with this disorder, but remain well as long as they continue to take this drug, because the entire pathogenesis of this leukemia was driven by this one mutation, and that knowledge has led to new therapeutics. In our own group, we've worked on a variety of diseases. One of these has been hypertension. Hypertension is a major cause contributor to heart attack and stroke. It affects 1 billion people worldwide and contributes to more than 12 million deaths every year owing to heart attacks and strokes arising from hypertension, and its pathogenesis has been largely unknown. We've scoured the world looking for individuals with the highest and lowest forms of blood pressures compatible with survival, and have used this to identify the fundamental pathways that contribute to hypertension that are relevant not just to these rare patients, but to the general population. And it turns out that there's one final pathway that is regulated, that regulates blood pressure in humans, and that is how the kidney handles salt. So every day we have 1 million nephrons in our kidneys, and we filter about 1.5 kilos of salt and need to reabsorb all but about 1% of the filtered load, and that's done by a series of transporters, channels, and exchangers. And we've identified mutations that cause these extreme forms of high blood pressure, shown in red, or low blood pressure, shown in blue. And impressively, all of the single gene forms that drive blood pressure to these extremes converge on this final pathway that, in which high blood pressure is caused by mutations that increase salt reabsorption, low blood pressure is caused by mutations that impair salt reabsorption. And the importance of this is has identified new pathways to try to intervene with both preventive and therapeutic strategies. And so now more than 30 countries around the world have programs to reduce dietary salt, to reduce the incidence of hypertension and heart disease. The Institute of Medicine in the United States has explicitly proposed a way to do this by reducing the level of salt in processed foods and restaurant foods. And this work has identified rational treatments based on these patients with rare mutations that drive blood pressures to the extremes. So the collective impact of this work is that we now have about 3,000 genes with mutations that have been identified that cause human traits. And this has transformed our understanding of the biology of every organ system in humans. But this is not the end of the story. So the cost of DNA sequencing has been dropping dramatically. So we're proud of pointing to Moore's law in computation, where the cost of computer memory doubles at the same cost about every 18 months. Well, the cost of DNA sequencing has come down. When we started sequencing the human genome in 1998, it cost $100,000 to sequence 1 million bases of DNA. Today it costs about a penny to sequence 1 million bases of DNA. So this spectacular reduction in cost of DNA sequencing has suggested that we ought to be able to learn more from sequencing genomes at a greater rate. So this next generation of DNA sequencing, each machine DNA sequencer can produce about 500 billion bases of DNA sequence per day, enough to provide complete sequence of 5 human genomes. It's done by DNA synthesis using these 4 different colors for each of the 4 different bases. So it's done on tiny slides where we can put 300 million spots, each from a clone of DNA, add 1 base at a time, and the color of the base that is identified by a microscope is capable of telling us which base has been added at each spot. And as each spot grows, we can determine the sequence of DNA. And this is one of the slides that that we do these sequence reactions on. And as you can see, they're tiny and have tiny little channels on them in which we introduce the DNA and do the sequencing on the machines that I have shown you. I'll be glad to pass this around. Please don't take it out of the container because it's very sharp. But on this, we can sequence hundreds of millions of bases of DNA on a daily basis. To do this at scale, out on the West Campus, just down the road from here, we've set up the Yale Center for Genome Analysis, where we run the DNA sequencers. We have computer architecture to handle the data. As you can imagine, the volume of the data is extraordinary, and we need very large data storage and high-performance computing capabilities to manage the data. And over the last several years, we've sequenced the protein coding regions of more than 40,000 people to study diverse disorders of human biology. So where should we be using this technology? Well, we know that there are about 20,000 protein coding genes. We only know what happens when about 3,000 of these genes are mutated. And as a consequence, there's a lot that remains to be done. We expect that every human gene, when mutated, except with a few exceptions of highly repeated gene families, will have consequences when these genes are mutated. And so we developed technology to allow us to selectively sequence just the 1% of the genome that has that encodes the protein coding parts of the genome, the business end of the genome, if you will. And so how are we using this technology today? Well, the first thing we did was begin deploying this to make diagnoses in people who were severely ill who did not have a known diagnosis. And this gives the example of the first clinical diagnosis by this genome-level sequencing. This was an individual who had failure to thrive, volume depletion. It was unknown what his diagnosis was, and we were able to show that he had a mutation in a gene that impaired his ability to absorb the ion chloride in his colon. We've gone on to use this to identify a number of new diseases that were not previously recognized. For example, this individual had high fever and respiratory failure and bleeding from a number of sources of unknown cause. It was thought that he might have an infectious cause, and we ended up by DNA sequencing showing that he had a mutation that caused his disease, that defined a disease that was not previously recognized. It's in a gene that is normally involved in the bacterial response to inflammation. And he had a mutation that caused that to be kicked off by other events such as physical or emotional stress. And as a consequence, this led to a massive outpouring of proteins that caused his fever and inflammation. And he ultimately recovered by being put on high-dose immunosuppression. And there are medications that will suppress the inflammatory molecules that he is making on a chronic basis. and can now be treated. And there are other families like him that have since been described around the world. We similarly identified mutations that cause a variety of tumors. Some of these tumors actually make hormones, and we showed that a common form of severe hypertension caused by tumors that make the hormone aldosterone are caused by mutations in an ion channel that cause the channel to now, rather than only conduct an ion like potassium, can also conduct an ion such as sodium, and that end up causing the cells to behave abnormally, leading to the constitutive production of this hormone and leading to severe hypertension. And these tumors occur in several million people around the world. And then more recently, we've recognized that we can use this technology to identify diseases that are genetic that we had never really been sure before were genetic. So for example, a range of congenital diseases such as congenital heart disease turn out to commonly be caused by mutations that are absent in their parents but are present in their offspring. So severe forms of congenital heart disease require correction early in life to sustain life. And we've shown that about 10% of these cases have new mutations that are absent in the parents but are present in the child. And a class of genes that are mutated most frequently are genes that are involved in as getting back to our earlier point, in wrapping chromosomes, DNA into chromosomes inside cells. And there are a number of modifications of these proteins that are involved in opening up the chromosomes to allow active transcription of the genes that are contained in those segments. And mutations that are commonly being found to cause congenital heart disease are mutations that alter the process of the modification of the chromatin in which DNA is wrapped inside cells. And this was a quite surprising finding that has come in the last couple of years. Importantly, this work is leading to new therapeutics. So in the case of cancer, we know that cancer is commonly a disease of somatic mutation, mutations that are absent in our genomes when we're born, but become mutated during the course of our lifetime, and these are what are driving the development of these cancers. So we now recognize, I told you about chronic myelogenous leukemia. There are mutations in the epidermal growth factor receptor in patients with lung cancer, for which there are now drugs that target the specific mutation, and there are other examples shown here along those lines. Similarly, targets for new therapeutics for pain, for cholesterol, for sleep, for Alzheimer's disease, for osteoporosis, have come from the identification of rare mutations that cause extreme forms of bone abnormalities or early onset of Alzheimer's disease. So I want to emphasize this link between finding new insights into therapeutic targets from identifying rare patients with severe forms of disease or health. So this is being rolled out on a national and international scale. The NIH is supporting Yale, Johns Hopkins, University of Washington, in an effort to identify the consequence of mutation of every gene in the human genome. These are going to not just define the consequence of these rare mutations, but will identify therapeutic targets for new approaches to therapeutic intervention in the general population. And then most recently, President Obama, recognizing the power of this approach in his State of the Union address this year, announced the Precision Medicine Initiative. And this initiative is to try to develop even further this link between genomes, mutations, common variations, human traits, and disease outcomes. This led to, just thinking about this for a moment, the path to preventing heart disease, where we've made so much progress over the last 75 years, started with an epidemiologic study in 1948 in which we followed several thousand members of the Framingham community in Massachusetts and identified which factors years later were the factors that predicted the development of heart disease in individuals. And that led to the recognition of smoking, high cholesterol, and hypertension as the major risk factors for heart disease. And we now have shown that if we reduce smoking, lower cholesterol and lower blood pressure, this has profound effects to reduce the incidence of heart disease. Well, the goals of the Precision Medicine Initiative are to do the same for every other disease. And the goal is to enroll more than 1 million Americans to follow them longitudinally and identify what factors at the time of enrollment are predictive of development of virtually every human disease 5, 10, and 20 years later. So this will be a large-scale longitudinal cohort study that will combine genomic technologies, metabolite measurement of many metabolites, measurement of the development of disease, and measurement of disease outcome. It will be a very large project coordinated across many sites across the United States. I had the privilege, as Kurt noted, of co-chairing the planning process for this, and we just finished this task, and you'll be hearing more about this as time goes on and this project begins to get rolled out. So we have as a goal then to determine the consequence of mutation of all genes in the human genome and to use this to identify genes and pathways for the greatest potential for development of new diagnostics and therapeutics. How we're going to use DNA sequencing going forward is a major question. I think there's little question that we'll be using it to understand the causes of cancer in individual patients. And over at Yale New Haven Hospital today, we're routinely sequencing the genomes of cancers to determine what are the specific driver mutations in each cancer. We ought to be able to do a much better job of newborn screening to identify at birth who has a particular disease that needs intervention at birth. In the state of Connecticut, we now screen every newborn for 40 different diseases. We'll be able to do a much better job of this with DNA sequencing. We'll be identifying clinical mysteries, as I've described. We'll be sequencing for prenatal diagnosis, which is currently done. And the question is, will we be doing genome sequencing of everyone at birth to determine all of our inherited susceptibilities to disease? There are potential pluses to this. There are also potential minuses. We're not very good at factoring the odds of a disease. So if I tell you your risk of developing schizophrenia may be increased by a factor of 2, well, that may sound terrible until you recognize that your baseline risk is 1%, and if you double that risk, it would only be 2%. So making sure that we are conveying this kind of information in ways that actually helps people rather than increases our neurosis about diseases that we are unlikely to ever get remains a major challenge in the field. And then lastly, development of new technology over the last several years has provided profound opportunities to develop new methods that might allow us to actually correct specific mutations. And this is a topic of its, should be a future topic for this session to discuss in another venue, but is one of profound potential implication for how we think about health and medicine for future generations. I think I'll stop there, and thank you very much for your attention.